Hey IB environmental students, welcome to Abiotic Factors topic 2.2. We're going to start by focusing in on the different types of systems that we're going to look at abiotic factors in. And the first system type is marine, which is the salty sea. And here you'll see listed a couple of different abiotic factors. For instance, salinity, which is the salt level, pH, which we know is acidity level, temperature, dissolved oxygen, which is really important for how much the fish are able to survive and breathe. How many waves are there? Remember, this is the big ocean or sea, and that's going to be related to currents. Turbidity is the amount of material that's keeping the sunlight from passing through. So like how sandy is the water, how dirty is the water. Nitrogen is going to be a lot related to how much fertilizer's in the water. Sometimes a little bit's okay, but a lot can be bad. And then recently we've been talking about toxins and metals. We're going to want to check for those in our water areas as well. Next is fresh water, which is our lakes, rivers, and streams. Now we're going to have some bit of a flow to our stream water. So we could put, for instance, a ping pong ball on a stream, and we can measure the rate it travels down a stream, and that'll give us a velocity. Once again, we can measure how acidic or basic the water is. Temperature, again, how much oxygen is available to the life in the water. Again, turbidity, again, nitrogen, and very similar, a lot of these other compounds that we might be nervous that could affect the wildlife there. Notice that we don't have anything that's related to salt water. There's not really waves or currents in a lake, river, or stream. Next would be our terrestrial areas. We're going to think about temperature in this area. We're going to think about how much light is getting to different areas there. The wind. Is there any dust in the air, any particular matter? That's going to also be related to smog or haze. Remember, if you've ever been on a city in a very gross day, sometimes you literally can't see past some of the car smoke matter. Slope, how steep is it? There's ways you can measure the angle of slopes. We're not necessarily going to do that, but if you are curious, please let me know. How much moisture's in the air and also in the soil, and that's going to be related to humidity. We can also look at a lot of factors specific to soil, since now we are terrestrial. We could look specifically at the pH of the soil. How much does water drain through the soil? Are there specific minerals like potassium, nitrogen in the soil? And once again, we can look at other nutrients or toxins or heavy metals in the soil or in the plant matter. Yep. Next, let's talk about the limiting factors that can occur in an ecosystem. These are, defined by IB, the biological necessities that an ecosystem cannot live without, whose presence in excess or of excess shortages can lead to adverse or negative effects in creatures. What's this really mean? Well, I want you to think about what potentially might be limiting that could negatively affect your future jar ecosystem. Think about, if you're terrestrial, what kind of things might affect the insects or the plants that keep them from having enough different necessities or nutrients. Think about in your aquatic ecosystem groups, maybe what might affect a snail or a shrimp from having everything that it needs. These are kind of tough, but go back to the previous parts of this podcast and think about those different factors and which things different organisms rely on as necessities the most. Now we're going to talk about some more methods. We're going to specifically focus on the Chesapeake Bay related methods. Some of these you might also be able to think about them in terrestrial systems as well. So the first thing we're going to really think about is pH as a factor and how we could measure it. Next we'll do temperature, and then we'll do turbidity and light. So when we think about pH methods, we could go back old school and do very simple methods, which is called litmus strips. And these strips change color depending on the pH of the liquid that they're in. If you were to measure soil, you can add some water to the soil and get the soil's pH as well. These methods are not very exact because You can't really see it very well, but each of the different colors on this strip tester kit, the colors are a difference of a 0.5 pH, which is a really large difference. To get more exact results, you need better methods, which would be through measuring with a probe. A probe is going to get a lot more significant figures, which is meaning that we could get into 2 decimal places, which is a much more accurate result and better methods. For temperature, we can use different types of simple probes to get even soil temperature or water temperature. This one almost looks like what you would use in your kitchen. It's not going to have very exact measurements, but once again, we have probes that could get very exact temperature measurements and changes, and even some changes over time. Next, we have turbidity, which again, if you don't remember, is how much light is going to penetrate through a liquid. And we could also think about this generally as an indicator of light. So this is called a Secchi disc. It has this funny black and white coloring to it. There's different meter measurements, usually on a rope connected to the disc, which is in water. A person would be standing above the water line, and they will look down and they will see, I see this funny black and white thing. If they can see it, then they keep letting the rope further down until they no longer can see this very distinct coloration in the bottom of the disc. If they can no longer see it, that means light's no longer allowing them to see it, and that they would mark that measurement down. Now, this is not necessarily the best method because the water in this area is changing quickly. There's probably some flow through currents or wave action. So this is not the best methods, but it's definitely cheap methods. A much more expensive method, again, is using a probe. Here we could put a little bit of water of a known turbidity in a vial, and then a unknown turbidity amount. So whatever my sample is, for instance, my jar ecosystem, for instance. And this probe is able to get a measurement comparing the known amount of light passing through a liquid to an unknown amount, and I can figure out how dirty or turbid my water sample is. It's pretty cool. We could also use for a more terrestrial system, this little circle here is a way of getting light sensor data directly to a handheld probe meter. This little fan device is also a cute little way to get a wind reading. So what I want you to now do is think about how you might be able to use these different methods for your jar ecosystem. Lastly, your turn. I want you to evaluate how maybe different abiotic factors might change over different locations and times. And to do this, please think about answering these questions under this objective. Explain how depth of soil or water might impact light availability and or temperature in your jar. So how much water or how much soil is that going to impact how much light or temperature you're going to have in whatever component of your jar system. Another really good question that you should have answers to next class is explain how time of day or year might impact different abiotic factors in your jar. Think about different abiotic factors and how they change during different times of day in order to answer this. Wonderful job, guys. See you next class.